Okay, welcome, my dear friends. Regarding of our CPGET 2025 free online coaching, today's our resource person, Dr. Bhagirath sir. Sir is going to explain one of the most important topic. Definitely one question comes from today's class, that is gene expression, especially the lac operon model. Which type of genes are involved, and the who given this model. This is most important. So, my dear friends, please listen carefully. And if you have any doubt regarding of our during the class, please clarify your doubts. Sir, please share your screen and start your session. So, please welcome.
I think screen is visible to everyone. Screen is visible, sir.
Yes, sir. Yes, it's visible, sir. Please proceed.
So, in the part of gene expression, the DNA, main genetic material containing number of genes, how these genes operate, its functions, and how it expressed, how it regulated during the protein synthesis process, how the genes are regulated at transcription level or translation level. It controlling mechanism is explained by some theories. So one of the theory which is linked to the lactose metabolism. So that theory name we can call as OPERON or lac operon concept. So in this concept, we can explain about what is meant by lac operon and what type of genes are present on the DNA and how it regulated and what are the positive and negative controlling mechanisms. These are the things we can explain one by one. So in an organism, different genes which are meant for the synthesis of different type of proteins. So number of genes are present on the chromosome. So these genes are meant for the synthesis of different proteins. But all these proteins are not needed at one time. So whenever it is needed, those genes are responded or expressed immediately with the signal by giving a DNA small portion. So whatever the signal information present in the DNA, that is coded and it is expressed by the synthesis of proteins. So however, some genes are constantly transcribed and expressed in all cells and all times. So why? Because their products are constantly needed in all cells. A cell, it maintaining its metabolism or the shape or the coordination, whatever it may be. If some cells always need few genes and it continuously transcribed and expressed in all type of cells and all times. So such genes are necessary to work continuously by the cell. Such type of genes are called as constitutive genes or housekeeping genes. So, but these genes are expressed only in some type of cells or on special occasion. That means on the DNA we can see number of genes, but few genes are expressed only in some type of cells. And on some special occasions or in some special metabolisms only. Such type of genes are called as non-constitutive genes or luxury genes. What is these differences between these two type of genes? Housekeeping genes are necessary to every cell to maintain its structure and shape and coordination with other cells. These cells are necessary and always expressed. But in case few genes, at whenever it needs, for example, A protein is need for a particular metabolism, then immediately the A protein responding genes are activated. The activated genes giving it undergo transcription and translation. The necessary protein molecule is produced. So those genes at that time only expressed. Remaining times the genes are in the off position. That means the control of gene expression or protein synthesis is called gene regulation. It is the process of turning genes on and off mechanism. Ante whenever it needs a protein molecule, the corresponding genes on the DNA will come to the on position. But a protein is not need or necessary at that time. Those genes are in off condition. So on and off mechanism is seen in the controlling or regulation of the gene expression. We can see with the help of OPERON concept. So that is the main thing you can observe here. So in prokaryotes, particularly the protein synthesis or gene expression is controlled at transcription level. At transcription level, the regulation may be negative or positive. The controlling mechanism is negative or positive. In negative regulation, an inhibitor or a repressor protein is present in the cell. This repressor protein is binding of the operator gene, and it prevents or suppresses the transcription of relevant genes. So what are the genes which are involved in the transcription, which are prevent or suppressed by binding the repressor protein at operator region? So this is one thing, negative regulation. That means here the transcription process is stops and we can't get a protein molecule. So in another way, positive regulation. In positive regulation, an inducer molecule is activates. The promoter is initiated and transcription of mRNA begins. In positive regulation, a inducer molecule is activates. Okay. The promoter region on the DNA, which starts and initiate the transcription of mRNA process. A negatively regulated system, in the first case, we can explain about negative regulation. The negatively regulated system may be either inducible or repressible. Inducible or repressible means what? Inducible or repressible? 1 minute. It is. Where is that? 1 minute. I think screen is visible. Hello. Okay. Inducible or repressible system. In inducible gene regulation, the structural gene or genes induced. That means the genes are switch on position to produce mRNA molecule. That is called inducible system. The substance which induces the gene for a protein synthesis is known as inducer. A metabolite or a substance which induces the gene for the protein synthesis, that particular substance is called as inducer. The gene complex responsible for the synthesis of indivisible enzymes is called as indivisible system. That is nothing but we can explain about with the help of Lac-Operon concept. So another example for the repressible system, it is negatively controlled or regulated, that is called TRP operon. Tryptophan operon concept is good example for repressible system. Next. Okay. So operon is nothing but it is a closely associated a group of cluster genes and which collectively regulate the protein synthesis process. So in another way we can explain like this: operon is a operational unit which can be defined as a cluster of closely associated linked genes on the chromosomes and transcribed together. It is a group of closely linked structural genes and associated control gene which regulate the metabolic activity. So the classical operon model was formulated by Jacob and Monod in 1961. These both scientists are postulated this classical operon model. Francis Jacob and Jacques Monod, both the scientists in 1961. So operon is nothing but in a simple way we can explain like this: it is a group of control genes and regulatory genes. The control genes having regulator gene, promoter gene, operator gene, along with structural genes. So promoter is that on the DNA molecule, a gene region on DNA where RNA polymerase binds to start the transcription. So the RNA polymerase where it binds on the DNA, that region we can call as promoter. The promoter gene we can encode it with R. Operator gene, that is O, region on DNA that controls access of RNA polymerase to genes, can activate or repress transcription. So operator is a main control point whether the RNA polymerase bind operator or not. It will be activated, sometimes it will be suppressed. So the structural genes are nothing but is functional cistrons. The functional cistrons are to be transcribed by RNA polymerase. After attachment of RNA polymerase enzyme, the structural genes are undergo transcription process and it produce respective enzymes. So the lactose operon designated as Lac-Operon. Lactose operon is nothing but it is associated with lactose metabolism in Escherichia coli bacteria. The Lac-Operon codes for enzymes involved in the catabolism of lactose. So lactose is a disaccharide sugar which is made up of with glucose and galactose. Both the sugars are linked and it is a disaccharide sugar named as lactose. It is indivisible operon since the presence of lactose induces the operon to switch on. So when the cell media containing lactose, definitely it induces the operon to switch on the genes. So indivisible operon since the presence of lactose. This lactose metabolism, when lactose molecule is available in the cell media, induce the operon to switch on the genes. So two kinds of regulatory proteins are there. What are the two kinds of regulatory proteins? One is activators Another one is repressors. Second one is repressors. Activators means what? A transcriptional activator, it is a protein that increases transcription of a gene or set of genes. So most of the activators are DNA binding proteins that bind to promoter. Most activators are DNA binding proteins which are bind to the promoter. The DNA site bound by the activator. Whenever the DNA site, that means promoter by the activator, is referred to as activator site. In the DNA molecule, where the location exactly bound by the activator, that position or location is called as activator site also. So example, the catabolite activator protein, that is CAP, is also known as CAAMP repressor protein. So it activates the transcription at the lag operon of the bacteria. So when a catabolite activator protein combines with CAMP, CAMP means cyclic adenosine monophosphate, it form a complex. This complex binds to the promoter, which starts the initiate the transcription process in the lag operon. Later we can explain in detail. Repressors. Repressor is also, it is also one of the DNA or RNA binding protein that inhibits the expression of one or more genes by binding to the operator. See here, where the operator region is there on the DNA molecule. A repressor is nothing but it is a DNA or a RNA binding protein that inhibits the expression of genes by binding to the operator. So in most of the cases, DNA binding repressor blocks the attachment of RNA polymerase to the promoter. So one of the protein, that is DNA binding protein, it acts as a repressor, blocks the attachment of RNA polymerase. And when RNA polymerase attached to the promote, it initiates the transcription process. But that process is blocked by the repressor. In the same way, this type of preventing of gene action or inhibit the gene expression at which level? Transcription level. So that's why it prevents the transcription of the genes into mRNA. So the DNA binding protein, which can act as a repressor, it preventing the transcription of the genes into mRNA. If a repressor is a RNA binding repressor, it binds to the mRNA and prevents the translation of the mRNA into the protein. And the prevention process is done at two levels. One is transcription level, another one is translation level. Translation level, the repressor is a RNA binding protein which binds to the mRNA and prevents the translation of the mRNA into protein. This blocking of expression is called repression. Repression, induction. So these are the two phenomena seen in the lag operon. So this is the lag operon model. In this lag operon model, operon is consisting structural genes, operator gene, regulator gene. These three kinds of genes, cluster of linked genes associated, and it regulate the metabolic pathway. Or see here, this is one is promoter region. I is the regulator gene. See, I is the regulator gene. O is the operator gene. Z, Y, A. These three genes are called as functional cisterns, or nothing but is called as structural genes also. These structural genes are involved in the transcription process. So promoter region, exactly in this region, the RNA polymerase attached and initiate the process. Is it clear? So these three structural genes represent the three enzymes. The cistern Z it represents the beta-galactosidase enzyme. And lac Y or cistron Y, it represents the galactose permease. And cistron A or lac A gene, which represents the thiogalactoside transestylase. So these are the three type of enzymes are synthesized with the help of three representative structural genes. So operator gene, promoter gene, regulator gene. So this is the cluster of these, all the genes are constitutively called as operon. So for example, in the cell media or in the cell cytoplasm, lactose molecule is absent at that time. In absence of lactose, how the gene expression takes place. So the regulatory gene which produce a protein that is called as repressor. This repressor protein, in the absence of inducer or lactose, which directly binds to the operator gene and block the RNA polymerase. So that's why no RNA made or no transcription process here. So when lactose is absent, repressor active and operon genes are in off condition. So that's why we can't expect three type of enzymes from transcription through the transcription process. So in absence of lactose, what happened? The repressor protein directly binds to the operator and block the attachment of RNA polymerase. So that's why we can't expect the transcription process furtherly. So if E. coli cell contains cytoplasm containing lactose molecules in the cytoplasm, definitely how the genes are expressed and how these genes are come to on position. So here lac I, that is very well, that is regulator gene which produce a protein that is repressor protein. When the E. coli cell cytoplasm containing inducer is nothing but here the lactose molecule. The another form of lactose molecule you can call as allolactose, which act like a inducer and which bind to the repressor, it become inactive. It change its conformational changes also in the repressor protein. That's why there is no chance repressor never binds to the promoter region. So operator region. So in the presence of inducer or lactose molecule which binds the repressor and it become inactive, that's why there is no chance of repressor protein binds to the operator. So the RNA polymerase enzyme ready and it attached to this region, initiate the process transcription. The lac Z, lac Y, lac A cistrons are ready to transcribe and undergo transcription process. Then translation, it gives three type of enzymes: beta-galactosidase, permease, transestylase. Is it clear? So what is meant by negative control of lac operon? So lac repressor is synthesized through the activity of lac I gene called the regulatory gene. The regulatory gene is responsible for the synthesis of lac repressor. This repressor is an allosteric protein, a protein whose shape is changed when it binds to a particular molecule. It binds the operator site, it binds the lactose sugar. So in the absence of inducer, what happened? DNA binding site of repressor is functional. In the absence of inducer, DNA binding site of repressor is functional. So the repressor protein binds to the DNA at the operator site of the lac locus and blocks the transcription of LAC genes by RNA polymerase. So that's why the LAC enzyme synthesis is inhibited. That is complete negative control. Negative control means the LAC operon genes are in off position due to the repressor protein. Positive control of LAC operon. So it is an additional regulatory mechanism which allow the LAC operon to sense the presence of glucose, an alternative preferred energy source of to lactose. Some cases lactose percentage or lactose content is very low and glucose is present in the cytoplasm. In that condition, if glucose and lactose are both present, cells will use up the glucose first. How? The presence of glucose in the cells switch off the LAC operon by mechanism called catabolite repression. So because when presence of glucose in the cell, how the switch of the LAC operon genes by a mechanism that is called as catabolite repression, which involves a protein called catabolite activator protein. So in the catabolite repression process, involve a protein molecule that is known as CAP, catabolite activator protein. CAP only binds in the presence of derivative of ATP. So in the earlier I said CAP binds with CAMP. CAMP means cyclic adenosine monophosphate. So when CAP binds to cyclic monophosphate, it forms a complex. This CAP binds to the DNA sequence upstream of the LAC promoter and enhance the binding of the RNA polymerase. So transcription of the operon is enhanced. How? CAP only binds in the presence of derivative, that is ATP called cyclic adenosine monophosphate. CAP binds to DNA sequence upstream of the LAC promoter and enhance the binding of the RNA polymerase. Transcription of the operon is enhanced. So CAMP levels are influenced by glucose. It depending upon the cyclic adenosine monophosphate levels. The enzyme adenylate cyclase catalyze the formation of CAMP and it's inhibited by glucose. When glucose is available in the cell, adenylate cyclase is inhibited and CAMP levels are low. The CAMP it derived with the help of adenylate cyclase. So when glucose is available in the cell, this enzyme inhibited and CAMP levels are very low. Under these conditions, CAP does not bind upstream of promoter and the LAC operon is transcribed at a very low level also. So if the presence of the glucose, it is also regulated and the CAMP derived by the catalyzing adenylate cyclase enzyme. This enzyme inhibits the process of formation of CAMP when it's low level. The never CAP does not bind to the promoter region. That's why the LAC operon genes are in switch on and transcribed at a very low level also. When glucose is low, adenylate cyclase is not inhibited, CAMP is higher and CAP binds to the increasing the level of transcription. If lactose glucose are present together, the LAC operon will only transcribe at low level. That means in the presence of glucose, in the presence of lactose, in the presence of both sugars, how this gene regulation takes place, we can explain with this CAP and cyclic adenosine monophosphate levels, and it regulated how it binds to the promoter, in which condition it binds to the promoter, and how the transcription process begins like that. So for example, only lactose molecule is available or not in the E. coli cell. For example, this is the presence of lactose in human gut, the Escherichia coli. the operon genes are on. So if the lactose is absent, the operon genes are off condition. So repressor from lag gene sits on the operator and block the transcription when there is no lactose in the E. coli cell. If lactose is present in another form, that is allolactose, when allolactose binds to the repressor, automatically the repressor inactivate. Repressor cannot bind to the DNA. That's why our promoter region is attached with RNA polymerase and genes are expressed. This mechanism is, we can compare our analogy with fan on and off mechanism. So when fan off, like lack operon, power supply is on, but a knob is there. The knob is act like a operator. If knob in off condition, there is no airflow. That's why no enzymes are produced in the cell. A fan is on position, like lack operon on. So a user with key, inducer comes and remove the block repressor. Now you can turn the knob into on position, fan starts the moving and airflow comes. So that is the analogy with what is that lack operon mechanism. So in this analogy, we can explain like this: component, power supply is there, electricity is available. Here, lack operon, RNA polymerase machinery is readily available for the transcription process. Here, knob, switch on or off position. It controlled by whether fan is on or off. In the same way, the operator region where repressor binds. If inducer is present, there is no chance of repressor comes here. So that is like fan on and off mechanism. Here operator region is whether repressor bind or not. Fan spins to cool air. Here structural genes are ready to construct or synthesis the enzymes. Block on knob stops knob from turning on. Here repressor the protein. User with a key. Remove the block to allow the knob, and inducer is ready, and the repressor become inactive. So this is the lack operon concept, which postulated by the Jacob and Monod. Hello? Hello? Hello, sir?
Regarding of this class, my dear friends, if you have any doubts, please ask our respected Bhagirath sir, because of it is the most important topic, and sir has clearly explained about the operon model. Actually, operon model, it is very closely related to the gene expression, how to control the transcription and translation process among the physiological functions in the molecular biology and the animals, plants, including prokaryotes. From prokaryote to the higher organism, all levels, it is controlled by this operon model. Jacob Monod has clearly explained about the lac operon model. Sarah has clearly explained how structural genes are involved, how repressor action is controlled uh, by the presence of the lactose or even the absence, and also the repressor operator functions sir has clearly explained. Thank you very much, sir. We're going to wind up the session.
Okay, sir. Thank you.